Buddy, thank you for joining us for the November issue of the seminar on physical genomics. My name is Benjamin Keene. I'm the Senior Program Administrator at CPGE here at Northwestern. We're really excited to have our invited speaker today, Dr. Jan Lammerding, who's a professor of biomedical engineering at Cornell University. Professor Lammerding's lab conducts pioneering research on nuclear mechanobiology, investigating how the mechanical properties of the cell nucleus modulate cellular functions and how physical forces acting on the nucleus affected structure and processes, with important applications in muscular dystrophy, heart disease, and cancer. Dr. Lammerding has published over 100 peer-reviewed articles, including in journals such as Nature, Science, and PNAS. And his research is supported by grants from NIH, National Science Foundation, the Leduc Foundation, and the Volkswagen Foundation. Dr. Lammerding's work has received prestigious awards, including a National Science Foundation Career Award and the Keith Porter Award of the American Society for Cell Biology. Professor Lammerding, thank you for taking the time to share your work with us today. A quick note for our audience, we have a Q&A session at the end of the talk, but you can also just type questions into the Zoom chat anytime when you think of them, and we can just address them during the Q&A. So don't hesitate to type those questions in. If you'd like to learn more about the research going on here at the Center for Physical Genomics and our training programs, you can visit our website at physicalgenomics.northwestern.edu. You can check out our video archive on YouTube or follow us on Twitter, X, Instagram, all those things. We record these talks and post them to our archive. So there's a good amount of content up there, and it's always growing. So anyway, at this point, Professor Lammerding, I can turn the floor over to you. Thank you once again for sharing your work with us.
Great. Thank you very much, Ben, for the kind introduction. And thank you for the opportunity to give us a chance to present some of our latest research here. And while I'm the person, I guess, presenting, I should point out this is really almost all of this the work by a fantastic postdoc in my lab, Julian Marvel, who's really just been driving this project, I think generated almost all of the data here. And he is on the job market now, so anybody who's looking to hire just fantastic new junior faculty, feel free to reach out to Julian and you'll be extremely happy with him. So my lab, we study three different research areas, and they all evolve around the nucleus and the mechanobiology of the nucleus. A lot of our early work, and we still continue to do it, is related to mutations in nuclear envelope proteins such as lamins or nesprins that result in muscular dystrophy and heart disease, where we're trying to understand what are the molecular mechanisms that go from a mutation in nuclear envelope protein to actually now muscular dystrophy or heart disease, and can we leverage some of these insights to develop novel therapeutic approaches. We do quite a bit of work also on nuclear mechanobiology in confined cell migration, trying to understand both how the physical properties of the nucleus, such as the size or the deformability of the nucleus, determines if a cell is able to migrate through tight spaces. And so we use microfluidic devices for this, but also study cells in vivo. And this is highly relevant, for example, for metastasis of cancer cells as well as immune cells. And importantly, we recognize that the nucleus is not just a passive obstacle that cells need to drag through these constrictions, but we have some very intriguing observations as well, where we see that deforming the nucleus in the process can result in changes to the chromatin organization, to the gene expression. And since we've already published some of the work on that, I decided today to focus more on our third research area, which is actually the overarching research area of our lab, is to really get a better understanding of the cell nucleus as a cellular mechanosensor. And so I think many of us are familiar with the fact that cells respond to mechanical cues, and they can translate mechanical cues into biochemical signals in a process that is called mechanotransduction. But much of the research has focused more on the cell surface, focal adhesion, the cytoskeleton, and we really are trying to understand to what degree then is the nucleus itself capable of sensing and responding to mechanical forces. Before we get there, and so today I'll just focus on this aspect of our research. Before we get there, I just want to just make sure, and I guess with this audience, it's probably I don't have to do a lot of convincing about why the nucleus is important and what we should know about the nucleus, but to make sure that everybody's on the same page. So we all know that the nucleus, of course, is very exciting because it contains the genomic information. But in many, I think, textbooks, and I think conceptually you often think of it still, the nucleus is just the bag of DNA that's floating around somewhere in the cell. And as long as it's just a confined compartment, that's all it takes. But it's really important to recognize that the nucleus is not just a bag of DNA, but it's actually tightly connected to the rest of the cell. And so we have I guess I'll talk about a little bit the nuclear lamina, but then primarily also the link complex. So we have these proteins that connect the inner nuclear membrane to the chromatin, but then also through these sun proteins and Nesprin proteins at the inner and outer nuclear membrane respectively, and they're engaging across the lumen. And so they're able to now connect from the inside of the nucleus to the outside. And these Nesprins on the outside, they bind to all major cytoskeletal filaments. So they can bind to actin filaments, to microtubules, and to intermediate filaments. And so therefore, you have now a physical connection all the way from the cytoskeleton, or even through the extracellular matrix to the cytoskeleton to the nucleus. And so forces are transmitted from the outside to the nucleus. And the question is, of course, well, do we just get deformations, or can that result in some biochemical signaling as well? And so to now get back onto the topic of the nucleus as the cellular mechanosensor, we know from extensive work over the last several decades that really this mechanotransduction, the ability of cells to sense and respond to the mechanical environment, is crucial for the maintenance of tissues, for development. And it's not just a few highly specialized cells that are maybe important for touch, but it's basically every single cell in your body responds and adapts to mechanical forces. And we see here, I think, a few examples, whether it's a beating cardiac myocyte, which exerts forces but also responds to forces. We know that muscle cells, if you go to the gym, if you exercise, your muscles will become stronger. If you spend time in bed or maybe on the International Space Station in microgravity, your muscles, your bone will become weaker because they're lacking the physical stimulation that they typically get. But we also see similar observations in cell migration, or for example, in blood vessels where the endothelial cells align in response to the shear stress. But as I said, much of the research in the past had been focused on what happens at the cell surface and how do, for example, integrant focal adhesions sense these mechanical forces. And so the canonical pictures, typically we have some mechanosensors, often at the cell surface or maybe at the cytoskeleton, that in response to mechanical stimulation, whether that's stretch or some force application, activate some signaling cascade. So they translate this physical stimulus into a signaling cascade, and then you have transcription factors that are now moving. They may be being phosphorylated or otherwise activated, and they're moving from the cytoplasm into the nuclear interior. In the nuclear interior, the transcription factors now act on the target genes, and they turn on the expression of these genes. And this leads then to the cellular response, which can occur on the order of maybe tens of minutes to hours, depending what the particular response is. And so it had often been speculated whether, in addition to these receptors at the cell surface, could the nucleus itself participate in mechanotransduction, in the sensing of forces. And there's been indication, and some of this goes back to when I was a graduate student 20 years ago, where we had shown that nuclear envelope proteins play a crucial role in mechanotransduction. So if you take a look at lamin deficient cells, which are lamins, we know if you delete them, you get a muscular dystrophy. And so we suspect that maybe these cells have defects in responding to mechanical cues. And so you can see how old the work is that we're still using northern blots to look at gene expression. And you can see if we take cells, in this case, these were mouse embryo fibroblasts that are either wild-type fibroblasts or cells where we've deleted lamin proteins. And if you subject them to cyclic mechanical stretch, about 4%, and then we can look at induction of known mechanoresponsive genes such as IEX1 or EGR1, you could see that wild-type cells, they respond with a strong activation of these mechanoresponsive genes, while the lamin deficient cells do not respond. So you basically get no signal here at all. And we've seen similar defects with amarin deficient cells as well. And so in many articles, you'll read that they said, well, that clearly shows that lamins are involved in mechanosensing. And I'd say I think we have to be very careful with that interpretation because we only see really that they're involved somewhere in the pathway. And clearly, if we delete lamins, we impair the overall response, but we don't know if this means that really that the nuclear envelope is involved in sensing mechanical signals, or whether it maybe does plays a role in processing these signals, but maybe more in a biochemical range, and that the signals themselves originate in the cytoskeleton or the cytoplasm or at the cell surface. And so we wanted to then just develop tools to study this in detail. And I should point out over the last several years, there have been increasing studies that looking at the role of the nucleus of the mechanosensors. Overall, you still find probably more review articles on that topic than actually primary research, but there have been several intriguing candidates emerging over the last few years, ranging from, for example, that tension in the plasma in the nuclear membrane can result in opening of the nuclear pores, which can facilitate import of transcription factors. The stretching of the membrane itself, coupled with calcium influx, can result in the recruitment of, for example, CPLA2 to the inner nuclear membrane, which can initiate signaling. There's been some reports that stretching of cells or applying forces can result in opening of chromatin, which could then result in the activation of genes. And there's also been several findings that implicate changes in nuclear and perinuclear actin polymerization in changing transcriptional responses. But again, for the most part, there's a lot of very open questions about this process. And so what we wanted to do is to see, can we use a very systematic approach now to study what role the nucleus plays in mechanotransduction? And we were able, I guess, to leverage some of our very ancient devices that we developed 20 years ago to stretch large numbers of cells. And back then they were developed to stretch millions of cells so we could get enough cells for a northern blot. But it turns out that millions of cells are really, really good as well to use some of the more modern genomics approaches. And so what we've been doing now, or when I say we, is I guess it's mostly Julian been doing, to develop this pipeline where we can take fibroblasts and apply cyclic biaxial strain to them, typically about 5%, which is low enough that we're not damaging the cells, but it's high enough that we really get a robust stimulatory response. And we apply the strain to these cells, and then we can either map, for example, polymerase to position. And so we're using a technique developed by John Lissy at Cornell, which is called PRO-seq, precision nuclear run-on assay, which allows us to really exactly map the position of polymerase to along the genome so that we can see nascent transcription rather than having to wait to produce RNA. We can use ATAC-seq just to map changes in the accessibility of chromatin. And we can also use, for example, Hi-C or micro-C, as well as other approaches to capture the chromatin context and chromatin conformation, which I'm not going to cover in too much detail today, which is work we've been doing in collaboration with Rachel McCord and Charles Danko's group. And I should point out, in our studies, we are really interested in detecting the earliest events because we're interested in the mechanosensing itself. And so we want to see what are the first genes that are being turned on, how fast do they come on, and not so much in terms of what are the long-term responses of mechanical stretch. And so we really focus on mechanical stimulation as short as 1 or 2 minutes. And typically the longest time points we're doing are about 30 minutes because we know that the earliest genes that are activated are typically transcription factors that then turn on additional genes and essentially create a second wave of transcription. And we really want to focus on the first wave of those genes that are being turned on directly in response to mechanical stretch. To provide a little bit more background on the PRO-seq, so the precision nuclear run-on assay, in case not everybody's familiar with it. So this was developed by John Lissy's group. It consists of permealizing the cells and then doing a nuclear run-on, so where you add nucleotides that are labeled with biotin. And this has a dual purpose with it. One, it allows us, based on the incorporation of the biotin now into the nascent RNA, just to pull out the nascent RNA so that we can really see what is being transcribed at the moment of the assay. The other purpose is that the biotin itself on the nucleotide will stall the polymerase. The polymerase is unable to process any further. So which means we really see the exact location at which the polymerase stood when we performed the experiment for the run-on assay. And then what we can do is we can enrich the biotinylated RNA, and then we can apply just relatively standard sequencing approaches. I guess it's a little bit more elaborate to prepare the libraries, but we can then just sequence now the nascent RNA. And so we can use that to map and get a clear picture where the polymerases are located. And what it looks like, this is typical data that you would see where we look at the gene body of one of these mechanoresponsive genes, EGR1, and we can see at 0 minutes, we have a lot of polymerase close to the promoters in the promoter-proximal region, but very few polymerases are in the gene body itself. And then as we're stretching the cells within 10 minutes, you can see we have a large amount of polymerases are now located in the gene body. And so they're actively transcribing the gene. And you can see we can then use similar tools that you'd use for general RNA-seq analysis. We can look, for example, at just map the different reads, look at changes in the number of reads in the gene body. We can see here, if you come do a principal component analysis, we get very nice clustering between the unstretched cells, then the cells stretch for 10 minutes, and the cells for 30 minutes. You can see, and you might ask, why do we see so much of the polymerase in these promoter-proximal regions? And is that kind of unexpected if these genes are initially not expressed? But that is not unexpected because it's now well recognized that what's called the polymerase pausing is a very common mechanism for gene regulation. So where you have polymerases are being recruited to a promoter, they'll start transcribing and then they pause within about 20 to 60 nucleotides from the promoter side. And then the polymerase essentially hangs out there and is just waiting for signals, which then result in the recruitment of elongation factors and the dissociation of pausing factors. And that allows then these genes to be very rapidly expressed. And so that's something that had been very well characterized for heat shock proteins by John Liss's lab, but it's now recognized that that plays a role for the regulation of many, many other genes as well. And so what happens now, kind of, if we apply stretch, so you can really see that this technique allows us to look at, even within 10 minutes, detect a rapid increase in the expression of many, many different genes. I think just I've shown here the EGR1 gene, which we've plotted here. You will see a lot of other transcription factors. And I think if you maybe recognize some of them, you'll see many of these are transcription factors that are typically immediate early genes. So they're transcription factors that are rapidly activated that then turn on additional genes as well. We can see if we compare, for example, a 10-minute time point with a 30-minute time point, that at 10 minutes, we already have an extremely strong induction of many, many genes. Some of them will just increase their expression further at 30 minutes. Others, they're already almost maxed out at 10 minutes already. But you do see that then additional genes come on as we continue to stretch cells. At 30 minutes, you find more genes that are being upregulated than what we see at 10-minute time point. Importantly, the nice part is about the PRO-seq analysis that provides not just information about which genes are upregulated, are induced, as you would get from a standard RNA-seq assay, but it allows us more insights into the dynamics as well. And I should point out, it is also much more sensitive because with RNA-seq, you typically have to wait long enough just to build up a sufficient pool of the newly transcribed RNA in order to detect it above the background of the existing pool of RNA. With PRO-seq, since we're only looking at the nascent RNA, so it's much easier and much more sensitive to detect chains of genes that are just being turned on. And because we're mapping the position of the polymerase, we don't even have to wait for the gene to be completely transcribed so that we actually have full messenger RNA there, but we can already map it while the gene is being transcribed. And this is something that becomes more apparent when you look at large genes. So the EGR1 or CFOS genes, these are relatively small genes at about 5 kb or less than 5 kb in size. So by 10 minutes, the polymerase has completely processed through the gene and the gene is fully transcribed. On the other hand, the NFAT-C2 gene, for example, which is a very large gene, you'll see here it's about just 80 kilobases long. At 0 minutes, we see very little polymerase activity within the gene. At 10 minutes, we can now see that the polymerase has started to actively transcribe the gene. So we've been transcribing about 25 to 30 kilobases of the gene at the 10-minute time point. And then if we continue to stretch the cells for 30 minutes, you'll now see we have now the polymerase, kind of the earliest polymerase has probably reached here about somewhere around 60 kilobases. You can really nicely see the progression of the polymerase through the gene body. If we were to do RNA-seq on just fully transcribed RNA, you wouldn't get anything until probably at least 30 minutes or longer, because this is how long it takes us to transcribe the entire gene. But the nice part is now, so based on the progression, we can also see how fast the polymerase progresses. It matches what we see, what had been previously described, that typically RNA polymerase 2 processes with about 2.4 to about at most 3 kilobases per minute, which I think matches in terms of what we see here within 10 minutes, and then the further increase. But it also means now, if we calculate backwards, if we know that polymerase 2 processes with about 2.4 to 3 kilobases per minute, and at 10 minutes, we have already progressed about 25 to 30 kilobases into the gene body. That means that the polymerase really must have started transcribing the gene almost immediately as soon as we turned on the mechanical stretch. And so that is something that is surprising, because I mentioned in the canonical mechanotransduction pathway, you typically have some signaling cascades at the cell surface, which result in some kinase activation. It results then in the phosphorylation of a transcription factor. That transcription factor needs to translocate from the cytoplasm to the nucleus. And typically that takes several minutes. And so if you look at classical studies that look at translocation of transcription factors, oftentimes you really only see increases in the transcription factors in the nucleus around 2 to 10 minutes after the stimulation. And so here we already see that the transcription must have already started within basically 1 or 2 minutes or even faster. And so that's really just one of the key findings that we've had, that transcriptional elongation must have started almost immediately upon the stretch activation. And we've done some more preliminary studies now, and I think in these preliminary studies, some of them, the read counts are a little bit lower than what we'd have in the other studies, so it looks a little bit sparse. But overall, you can clearly see it in the heat maps. We find when we map even at 2 minutes that there's several genes, particularly, for example, the phosphorus-B genes, but we see substantial transcriptional activity already at 2 minutes. You'll saw, I think, the NFAT-C2 gene that I showed before. We can see some activation. The ATF3 gene, which I think I showed the plots here. Again, those all show substantial transcriptional activity already within 2 minutes, so much, much faster than what we expect that the canonical pathways are able to move through. And so this is something that we are very excited about now, to trying to understand how are these genes being activated so rapidly. So we can gain some insights about some of the regulation directly from the PRO-seq studies. And so the nice part is, with PRO-seq, you're not only learning about transcriptional activity within the gene body and to see which genes are being turned on, but we can also monitor transcriptional activity at the transcriptional regulatory elements, so particularly at promoters and enhancers. And so it's known that promoters, you can find transcriptional activity at the promoter itself. And then at enhancers, you can see the bidirectional activity, so riding in both direction of the enhancer. And so you can identify these signatures to determine now which enhancers and which promoters are active in response to mechanical stretch. And so we can then perform a predictive modeling and just using a motif analysis of these transcriptional regulatory elements that we see activated in response to mechanical stretch. And so we identify several in terms of known mechanoresponsive transcription factors, such as the FOX family transcription factors, SOX. But we've also seen, for example, that P53, which is typically thought of more as a DNA damage response gene, that is being activated very rapidly in response to mechanical stretch as well. And so we can then map, for example, also to see which of the differentially expressed genes that we have identified in our studies come closest or are located nearby some of these transcriptional regulatory elements that we've identified. And we see that many of those match in terms of up with the genes that we found, that, for example, we have P53 can potentially act on the FOS-B and FOS genes, that we have NFAT-C2 is being responsive likely to several of these transcriptional regulatory elements. We can overall kind of just to summarize some of these data. So based on the transcription factor motif analysis, as well as ingenuity pathway analysis and other analysis, they point to potential transcription factors and regulatory pathways that are responsible for this very rapid activation of genes. And so these transcription factors include, for example, the NF-kappa B pathways, MAP kinase pathways, P53, and the FOXO pathway, which again, many of these pathways, such as NF-kappa B, MAP kinase, are very well established mechanoresponsive pathways. For NF-kappa B, that's I think true as well. For P53, there have been some findings, but that was still a little bit surprising to us that that is really being activated so rapidly in response to mechanical stretch. And we are now in the process of performing ChIP-seq and ChIP-PCR as well as ChIP-exo to confirm some of the binding of these transcription factors indeed to the target motifs of these specific genes. And so one interesting question was also, do we see differences between, for example, different cell types or when we apply larger strains? And so we've taken a closer look at some of the analysis of our differentially expressed genes and then the protein-protein interaction network. And so what we've seen is really that most of the genes that come on very, very rapidly are immediately early genes. So they're transcription factors that are associated often with cellular stress responses. They're all in a very tightly interconnected protein network, except I guess NFAT-C2, which was another gene that we found that stands a little bit outside. But again, you'll see a lot of connectivity between them. And so this is work done in human skin fibroblasts that we've stretched. These are some of the pathways that come on that have been associated with the activation of these genes. And so these are some of the upstream regulatory factors that are predicted by the ingenuity pathway analysis, where again, you'll see SRF, which this is a classical mechanoresponsive genes, again, the FOX transcription factors, NF-kappa B. So it is kind of, we keep seeing the same suspects that are involved in the regulation over and over again, regardless of in terms of which analysis we perform. We've wanted to see whether if we now stretch different cell types, or in this case, even a different species, whether we see similar responses in the sense, do different cells have maybe a canonical mechanosensing program? And so these are very preliminary experiments where we used primary mouse myoblasts that we then differentiated in vitro to form myotubes so that they fuse together for multinucleated myotubes that we can then subject to stretch. And then again, we use a very similar stretch protocol. We get very similar results. Again, we can identify in terms of just the network of the mechanically activated genes. And you'll see again, EGR1, FOS, FOSB, come on, ATF3. So we have a lot of the same genes being activated. And if we look at it, and this is still very preliminary, where I think some of the read data that we have from the myoblast is not quite of the same quality of what we have for the fibroblast, but we already see that there's substantial overlap between the transcriptomic signatures and particularly between these very rapidly responding genes. And so we really think that there's some fundamentally conserved genes that are across many different cell types, in this case, even across many different species, that are really activated by mechanical strain. And then those probably turn on downstream other signals, and the downstream effects will likely be highly cell type specific, where you can imagine EGR1 might turn on different genes in a fibroblast than in a myoblast. But it's really the fact that EGR1 and ATF3 and FOS are being so rapidly turned on that allows a cell to respond to these mechanical cues. And so one of the major questions then, of course, is this, well, what mediates this rapid gene induction? If, as I said just there, it seems to be that the canonical path is where a transcription factor needs to translocate all the way from the cytoplasm to the nucleus. That might be too slow to explain the rapid activation. And so the first thought we had, well, if something is really rapid, could it be that it's calcium? Because we know that calcium, the stretch-sensitive ion channels, such as the piezo channel and various TRPV channels that are located in the plasma membrane, in the nuclear membrane, in the ER, and work has been shown, for example, by Sarah Wickstrom's group, that these ion channels are responsive to mechanical stretch, and they respond very, very rapidly. So if you look at calcium signaling, you start stretching cells, and then within seconds, you get a strong calcium response. However, in the canonical pathways, typically, even though you can get a very rapid calcium response, it still then takes several minutes to go through, for example, calmodulin, because you still need some phosphorylation and some translocation events. But we wanted to see, well, what happens if we block calcium signaling, if we chelate calcium, and will that affect our mechanoresponses? And so what we can do is we can use chelators, and in this case, we use BAPTA-AM so that we can chelate intracellular calcium. We can perform the same experiments. However, what you see here, if we compare conditions, and this is for the 30-minute time point, but I think we have similar results for the 10-minute time points, and we're repeating it now, the 2-minute time points. We don't see too much of a change. So the same genes essentially that come on in the control conditions, even if we use BAPTA-AM to chelate the calcium away, pretty much most of the genes still turn on. So we can, I guess, see there's a few very subtle differences, but none of those were statistically significant. And so for the most part, our impression is that the rapid activation of the genes is not dependent on calcium signaling. We do see, if we look at the processing of the genes, that the processivity or the rate of elongation appears to slow down slightly. So in the cells that are treated with BAPTA-M or we chelated calcium, you'll see that the progression of the polymerase along the gene body is somewhat slower than what we see in the control cells, which would, to me, suggest that it's probably not so much an effect on the activation of the gene, but then for the polymerase II, in order to process the gene, you do probably need some calcium. If you have chelated calcium, that the processing is not as fast. But again, these are preliminary studies now that we are confirming now in additional experiments. The other mechanism that had been proposed, and there is just some work by Ning Wang, and I think I just saw, I think Andy Belmont here as well on it, that had pointed out to that there could also be potentially mechanically induced changes in the chromatin conformation. And so what these experiments, I think some of the earliest papers in 2016, there's been a number of follow-up papers, suggested that the mechanical forces that are transmitted from the cell surface through the cytoskeleton, through the link complex to chromatin can result in potentially unfolding of chromatin, so that you're now making some sites in chromatin accessible where you could now recruit transcription factors or polymerase, and that could result in the activation of gene expression, in particular in the very rapid activation, because mechanical stretch and mechanical conformational changes would be expected to happen on the order of seconds or even faster. And so in order to address that, we've performed experiments where we used our same experimental pipeline, stretching cells, but now instead of performing PRO-seq, perform ATAC-seq or OmniATAC, so where we can measure the accessibility or changes in the accessibility of chromatin. And we can do this genome-wide now, and we can do it either just at genome-wide, or then we can hone in and look at specifically at the genes that we know are activated in response to mechanical stretch. And what we found there was all of the genes that are being rapidly activated by mechanical stretch, such as EGL1, FOS, and FETC2, all of those already had a very open chromatin structure around the promoter. And we did not see any further changes really in response to mechanical activations. You'll see it, for example, the EGL1 gene or FOS gene here. If you look at the ATAC-seq plots, which indicate the accessibility, you'll see at the promoters, so at the start site, all of them are extremely accessible already, which corresponds very much with what we saw in the PRO-seq data, that we have polymerases there, so that these genes are already primed to be activated. So they're already open. They have the entire transcriptional complex already assembled, and then they're just waiting for the proper signal in order to start the transcriptional and the elongation itself. So if we look, for example, across all of the differentially expressed genes, and then map the changes in the transcriptional activity, in the accessibility of the promoter sites, and we essentially see no difference at all between before stretch and even after 30 minutes of stretch. So we really see no indication of changes in the accessibility of the chromatin at the promoter sites. And the ATAC-seq or the OmniATAC data also allowed us to try to gain further information about some of the transcription factors binding to these promoter sites, and what we could perform what's called then does transcription factor footprinting. And the idea is if you perform very high resolution ATAC-seq data, you can identify the presence of transcription factor binding to a specific motif, because where the transcription factor is bound, generally the larger region itself is accessible, but the transcription factor itself will block part of the genome region, part of the motif, because the transcription factor will now make it inaccessible. And what you'll see is what is then described as the footprint. So you'll see an area with very little accessibility flanked by areas of very high accessibility. And so that is then the footprint. And what you can then do, you can try to see, well, this motif here, that should be characteristic of specific transcription factors. And so we can do that as an additional tool to identify potential transcription factors that regulate our differentially expressed genes. And again, we'll see that, for example, for EGR1, some of the closest motifs we find are for NF-kappa B. We'll see June, so we'll see several of the MAP kinase factors, where again, it all points to that we see a lot of the very classically conserved pathways, such as NF-kappa B, MAP kinase, and that those are responsible for the rapid activation of these genes. What we don't know yet, though, is how do they do it so much faster than what we expected and then what the canonical pathways predict. So in order to just then summarize and conclude, so I think hopefully what I've been able to show you is that really that stretching cell causes expression of mechanoresponsive genes within less than 2 minutes, likely even faster, and that these are typically immediate early genes that encode transcription factors that then add on additional genes. And then the activation appears to be due to rapid pause release of Pol II that's already located at promoter-proximal regions. Although there's a few genes, and I forgot to point this out, I realized, for example, at the NFAT-C2 gene, that there we did not see a lot of pause polymerase II. Nonetheless, we saw this rapid activation, so there must be some mechanisms to really very, very rapidly recruit polymerase II to the promoter-proximal region and then immediately start the productive elongation. We identified several transcription factors and regulatory elements that are driving this rapid gene induction, with more work going on at the moment to really identify the regulation of these genes. And at the same time, we can already fairly firmly conclude that in our experiments, it is not regulated by changes in the chromatin accessibility, because the chromatin accessibility and configuration, we have this data from the high-C studies as well. They do not change substantially within these 30 minutes in response to the 5% strain application. So instead, we have genes that are already poised and primed for the rapid expression. There's still several open questions, and I think this is just research that we've only started relatively recently, so we're very excited about it, but we're also just intrigued and be happy to receive feedback and potential collaborations from others to see, for example, what regulates this pause release. How do you get that mechanical force application results in such a very quick release of the pause? Does mechanical strain, how does it achieve the recruitment of Pol II to those genes that did not have a lot of pause polymerase II present there, such as the NFAT-C2 genes? What determines which mechanism is used by specific genes? So we clearly see, and that was very intriguing to us as well, that different genes have very different profiles and seem to use different mechanisms, with many of them using pause release, but others different mechanism. And is that based on maybe which transcription factors they're using? Is it based on maybe their location within the genome, where they're located at the periphery of the nucleus or not? So those are all open questions. We are starting now to get into, are these effects dependent on nuclear deformation? Is it really sensing of the nucleus itself and you need to deform the nucleus to get this rapid activation? Or could there still be some mobile signals, biochemical signals, that are just transmitted much more rapidly through the cell than we had thought is possible? And so some of the experiments we're testing now is to really look at these proteins at the nuclear envelope, such as lamins or nesprins that form part of the link complex, to see what happens if we disrupt the link complex so that we are no longer transmitting forces from the cytoskeleton to the nucleus, and how will that affect this very rapid activation of these genes. With that, I'd just like to thank the people in the lab who've done all the work, so particularly Julian, but we've also had several contributions from other people in the lab, particular Anna and Julie have done a lot of work with it. I think Jacob here has been contributing to it. And so we're just increasing recruiting more and more people from the lab to help address these fascinating questions. And with that, I'd just like to thank our collaborators, particularly Charles Danko, John List for their work, our funding agency, but most of all, thank you all very much for your attention, and I'm happy to take any questions. Thank you.
Thank you so much. So as I said, feel free at this point if you want to turn off your mic and throw out a question, or we've got one in chat. I don't know if you want to just read that and answer.
So I can see, so Kathina asked, does the speed and intensity of response change between different genes and cell types? For example, do the pause-release based genes respond faster or stronger? That's something that we're characterizing no more systematically right now. There wasn't any obvious difference, which I would have thought as well that the pause-release genes would be much faster. And I should point out that the vast majority of the DEGs that we detect use the pause-release mechanism, but NFET-C2, which does not seem to use pause-release, is also coming on really fast. And the change, the kind of the log-fold change, does not seem to be really different between them. So it doesn't seem to be that there's a clear correlation between those. Between cell types, I think that's still relatively early, but that's, I think, where we're hoping to get more data. I'm also hoping to get more experiments done where we vary, for example, the magnitude of the stretch that we apply, just to see if are there maybe some, particularly if we disrupt the link complex, for example, are there some genes that maybe do not come on at low strain rates, but if we use larger strains that are being activated then. And then I think Andrew asked us, can we speculate anything about the mechanism by how these immediate early genes are responding? And I guess I can speculate. I guess I'll start off with the speculation. So initially we thought, oh, maybe it is that it is calcium related, which I think doesn't seem to be the case, and it doesn't seem to be the unfolding. We still trying to see, is it possible that at least some of the transcription factors are already in the nucleus, so they don't need to translocate? It would still require some mechanotranslated signal to then activate them. And that's something we're trying to see. Is there something at the nuclear envelope or something in the cytoplasm that can rapidly enter the nucleus that could act on them? Or is it possible with heat shock? We know, for example, that there can be that the conformational changes that are induced by heat shock can then result in the sequestering of other factors that then release transcription factors. And is that something that mechanical stress could similarly activate as well? But at this point, these are really all speculations that we're just trying to pursue. And the other question was related to my first question. Is there something special about the non-I genes that respond? That's something I suspect there is something special, but we don't know yet what is special about them. So that's what we are trying to do now, a more systematic analysis. So one student is trying to really look at a genome-wide through all of the genes that we find. Can we essentially put them into different categories in terms of these are pause-release based genes, these are non-pause based genes? What do these different subpopulations then have in common? So can we identify, are they either based on maybe transcriptional regulatory elements, based on the promoters, or maybe based on the location within the genome or within the nucleus? Are they at the periphery or not? Do they have anything in common there itself? So I think hopefully in a few months, I'll have more and more precise answers for these questions. And then let's see how Luna asked, is there any other protein that temporarily and immediately holds to maintain the nuclear integrity before the transcription of mechanoresponsive protein to counter the mechanostress, since the transcription translation will take several minutes? I should point out that in our case, as with a 5% strain, we don't think we're really damaging the nucleus. And so we really intentionally used a strain regime that is small, so that is sufficient to stimulate the cells, but it's not damaging them. So we do not see any indications of nuclear envelope rupture. We don't see DNA damage. We don't see cell death. We don't see changes in the permeability of the plasma membrane. So I think at this stress regime, I don't think that the cells really need to worry too much about having to protect the nucleus. It'll be interesting to see if we increase the stress, but we wanted to avoid confounding factors of that are incurred when you damage the cells, because as soon as you damage the cells, you're going to activate several other stress responses of the cells, which would likely use very different transcriptional regulatory pathways. And so we wanted to remove that as a confounding variable, and so therefore just use conditions that are well within the physiological non-damaging range. And then when I say systematic analysis, what approach do you want to use to dissect the pathway systematically? So I guess there's multiple approaches. Part of it is really now just looking at the genes, since we have the genome-wide information, trying to see if we look at now the activated genes that use, for example, pause release versus those that don't use pause release, what common characteristics do they have in terms of transcriptional regulatory elements, in terms of positioning within the nucleus, and is there maybe size of the gene? Are there other commonalities in between them? And so really just trying to do a systematic characterization there.
Can I ask you 2 questions?
Oh, good. Yes, please. And I'd rather hear the questions asked in person. It's always nicer than just purely reading them.
Yeah. So first of all, very cool research. I really
Thank
like how you combine mechanical engineering with gene expression analysis.
Thank you.
So my first question is about how this stretch is being modeled. So if I understand the video correctly, this stress is being modeled on and off in a cyclic fashion. I was wondering, because if we think about skin stretch as you were using fibroblasts, the immediate thing jumped to my mind is women undergoing pregnancy. So their belly is getting bigger and bigger over time. So I was wondering if you tested a continuous stretch versus this cyclic fashion, and if there are any differences.
I would expect that to be different. So we haven't tested it here, but there have been a lot of studies previously in general mechanotransduction, where they often looked at focal adhesion and others, and they typically find that cyclic stretch result in a much stronger activation, because I think the thought is that cells will adapt to a static stretch. So if you stretch them, the focal adhesion, the cytoskeleton will remodel. So after a while, what felt like stretch no longer feels like stretch. And I assume that's probably happens in pregnancy, other conditions as well. The process is slow enough that it provides stimulation cells where the cells have time to adapt to it. Well, if you apply a cyclic stretch, you continuously provide a new signal all the time, and that's why you get stronger responses itself. And so that's why we tried to use the stretch here. So in this case, the more kind of comparison that I think, and what we're interested in is, for example, just that what happens with muscle contractility, where you have very acute, very rapid activation itself. And I think that's where we've started now doing some experiments, collaboration with John Fry's lab at the University of Kentucky, using nerve stimulation of mice to electrically stimulate the muscles very briefly, and then perform similar analysis there as well, that we can really look at the very active response to it. Because again, with muscle, there's a lot of studies that study gene expression 30 minutes after exercise, or maybe an hour or a day after exercise. But of course, to really understand what is the activating mechanism, you want to look at it in terms of within minutes afterwards. And of course, that's typically much more difficult to do, to collect the RNA then.
Great. So my second question is related to the mechanism.
Mm-hmm.
So we happened to finish a project about 2 years ago. It's a completely different project, but seeing some very similar genes being induced. So that project was focused on the super elongation complex. So that complex contains the CDK9, and it's related to transcription elongation. And what we found, because we use human skin keratinocyte, but not fibroblasts, to do all the analysis. So what we found is when we disrupt the super elongation complex in primary skin keratinocytes, we see very rapid induction of genes such as ATF3,
Oh, cool.
what else, FOS, and a few other genes that are also highlighted in your list. And another thing we identified, so mechanism-wise, we found is there is a subset of super elongation complex that's bound to inactive CDK9. So the conclusion we had for that project was when CDK9 was converted from the inactive state to active state, so a simple kinase activity gene was sufficient to convert from pause to elongation status. And the signaling we found very close to where you demonstrate calcium, but it's not identical. You can see a subset of those respond to calcium, a subset of those also respond to DEG. So we found when we treat it by TPA, so it mimics DEG was sufficient to mimic the disruption of super elongation complex. So maybe
that's very cool. And
could be something if you want to throw in like a TPA and try in your stretch to see if it like blocks or overrides the elongation you saw.
That would be happy, and I'd be happy to follow up there as well, because I think that is kind of goes along similar pictures where we think as well that you really, so you have the elongation complex, and then you probably just the stretch will just perturb it either directly mechanically or just through then a signal that can come very quickly, can directly act on it and phosphorylate one of the components, and that is all sufficient to then just trigger the pause release. And so I think would to me makes the most sense.
Great.
Okay, thanks so much.
Of course.
I think we've got one longer question in chat.
Let's see if I can. Let's see. Cody asks, does the response have to be dependent on transcription fluxing into the nucleus? If TFs are already present in the nucleus and the increased strain transduced to the chromatin promoters that are near the surface of heterochromatic regions, could this be a mechanism that might be employed? This might explain the nearly instantaneous response of some mechanosensitive genes. Maybe early mechanosensitive genes are poised this way. And that's, I fully agree. So I think the process is too fast to get transcription factors that haven't been in the nucleus to get those to move into the nucleus and turn on. So I think it must be factors that are either potentially already even sitting at the promoter and then are just being held inactive through some mechanism, or that they're already at least at the periphery and other sites in the nucleus, and so they just need to localize within the nucleus itself. So I fully agree with this. And I guess the experience will then hopefully show as well whether we really need mechanical deformation of the nucleus or whether it is purely just a biochemical signal that can be rapidly released, maybe from the cell surface, maybe from the ER or from somewhere else. So why do genes change a lot after cell stretch, but chromatin accessibility shows no difference? I think the reason for that is because all of these genes are already accessible. So there's no need for them to change because they're already poised, they're ready to be transcribed. And on the other hand, genes that are not accessible, they will likely take much, much longer to open up. So they might be in the secondary response. I wouldn't be surprised if you looked at it after maybe an hour, 2 hours, that then eventually you'll get some more remodeling, and so you'll get pioneer factors in, and you'll start now opening up genes that were not accessible. But for any of these really rapid responders, that those are already accessible.